Okay, so you're welcome to my lecture series. And like I say, these lecture series are going to be brief. I'm going to segment them into different segments so that you're not going to come up with a video that is too long or the one that is too short. So I want to go right into the study we are looking at, BCH 201, which is known as the General Biochemistry 1. It is the first lecture you're going to be taking in biochemistry, and it's going to be talking about amino acids. Okay, now what are our objectives in this lecture series? One, you will know what amino acids are. You will know what the different structures of amino acids. You will know the names and their different structures. You will know about classification of amino acids. Now we look at amino acid chemistry, that is the reactions which involves amino acids. And we talk about protein and amino acid derivatives. So these are what is going to we are going to be looking at in this study. So this first series we just end with an introduction. Now, there is no way we can talk about amino acids without looking at proteins, because it is the amino acids are the building blocks of proteins. So we say that proteins mediate virtually every process that takes place in a cell, exhibiting an almost endless diversity and function. Now, proteins are everything in the cell. Okay, the functions of the cell is depending on what the proteins in that cell are doing. And we say that proteins are the most abundant biological macromolecules occurring in all the cells and parts of the cells. So there is no biological cell that does not have protein in it. Now, proteins are molecular instruments through which genetic information is expressed. For instance, whatever character you think you're picking up from your parents or a character that is being transferred is as a result of the proteins, because the DNA which carries this information in the gene is transcribed into RNA, and the RNA is transcribed into protein. So every expression in the gene is being translated and is expressed in protein. Now, all proteins, whether it is from the ancient line of bacteria or from the most complex form of life, are constructed from the same ubiquitous set of amino acids. There are just 20 of them. And these 20 amino acids, are covalently linked in a characteristics linear sequence. So we look at the sequence of amino acids and we'll see how they are linked. So any protein you see, it must contain 1, 2, 3 or as many of the amino acids that we have, but these amino acids are just 20 in order, so in number, and the only difference is the sequence and the number of the amino acids in the protein. Now, proteins are found in a wide range of sizes, and it's from a relatively small peptide like glutathione with just few amino acids, let's say residue, to huge polymer of molecular weight that runs in millions. What is most remarkable that cell can produce proteins with strikingly different properties and activities by just joining the same 20 amino acids, but in different combinations and sequence. I have said that. So you can find out that the building blocks of so many proteins, such as enzymes, hormones, antibodies, transporters, muscle fibers, the lens of the eye, feathers, webs, horns, milk proteins, antibodies, mushroom poisons, and myriad of other substances that have biological activities are all proteins, and they are all formed with just these 20 amino acids, but with different lengths and different combinations. So I think you just have to digest this introduction, and then we continue some other time.